am studying a hypothesis in Parkinson's disease, which is that the disease might actually start in the gut. And there's growing evidence that the pathology in Parkinson's patients actually appears in the gastrointestinal tract years before it appears in the brain. And so we're really interested in studying this gut-to-brain disease process, and that's what we've recently gotten a DoD award to study, and specifically we're looking at cognitive dysfunction. So it's not that well known that Parkinson's patients in later stages often develop dementia, and so we are studying this gut-to-brain disease process and asking whether or not that, initiating the disease in the gut can actually cause cognitive decline, and how. And so we're using a very small model organism called C. elegans, which is a tiny nematode worm. And these worms are surprisingly and remarkably similar to humans in the sense that they have many of the same neuron cell types. They have between 60 and 80% of human genes represented in the worm genome. So they're actually remarkably similar genetically, and we can use them for really large-scale experiments. So because they're a small system, they're fast, they age within a few weeks, and then only live for between 2 and 4 weeks, we can use them to very quickly do these age-related disease studies and manipulate a lot of genetic processes. We're going to be testing different treatment options. So this is a platform to be able to do very large-scale experiments and understand disease mechanisms and try and find treatments. This is a 2-year award. And so in these 2 years, our goal is to identify the effects of this gut-initiated disease on cognitive function. So we're testing learning and memory in the worms. And we're also going to be identifying how that disease process occurs and travels from the gut to the neurons of the worm, and test several different FDA-approved drugs for the potential that they might be promising candidates to repurpose to Parkinson's patients.